Welcome. I want to welcome everyone to the first of our Alzheimer's Disease Challenge webinar. My name is Mette Peters. I'm with Sage Bionetworks. Before we get started today, I just want to cover a few logistical points when it comes to this webinar. We're going to have a question and answer session at the end, and we'll have all participants on mute. The way that you will submit questions for the question and answer session is by writing them into the question section in your GoToMeeting webinar. We'll log all these questions and we'll answer them at the very end. We may not, in the case that we don't get to all of these questions, we will track all of them and we'll post them on the Alzheimer's Disease Forum, including the ones that we have answered during this webinar so that you have a log of everything that we covered today. I also want to make the point that this webinar itself is being recorded, and we will post the recording, including these slides that you're looking at right now, onto the wiki or the Alzheimer's disease site that we have generated. So you'll have access to all of this data following the completion of the webinar. Let me see, just 1 minute. I'm having some problems progressing. There we go. So what I would like to start off with is introduce Laura Mangravidi. Laura will talk a little bit about the incentives for participation in the webinar. I am then delighted to have Simon Lovestone here today that will give us an overview of the importance of this challenge and the importance of conducting these type of challenges when it comes to a complicated disease such as Alzheimer's. Laura, turn it over to you.
Great, thanks so much, Mette. Well, sorry, so there's a couple of us in the room, so we had a little echo. There we go. I just want to take a second to thank all of you for your interest in the AD Challenge. We're very pleased to have you joining us in this endeavor. You know, there's been lots of work going on behind the scenes on our side preparing this challenge for all of you, and we are very excited over the next coming months to see your efforts, to see whether you can come up with novel, interesting, and informative models to assess some of the questions we've been looking at here. So let's see. So as I'm sure many of you are aware of the DREAM challenges, and in the past we've had many successful challenges looking at all sorts of interesting questions, both more basic research questions and increasingly we're starting to look at questions in translational research. And so this challenge really constitutes one of our major efforts in that arena. We are really interested in taking cutting edge and emerging modeling techniques and testing whether we can use them to really advance the field towards improving clinical care, in this case in Alzheimer's disease. But we want this to be a rewarding for you as well as for us and for patients in Alzheimer's disease. And so we do have some incentives that we hope will encourage you to stay involved in the challenge throughout. So the top team for each of the sub-challenges will be awarded an invitation to co-author a manuscript with us that will both describe the challenge and will describe your winning model. We want to know about your strategy, and we are partnering with Nature Neuroscience for this challenge, and so this manuscript will be submitted there. In addition, we will invite you to come present your method at the 2015 International Biomedical Commons Congress, which is happening in Paris, France, in April 2015. This is a Congress which is sponsored by Sage Bionetworks. It has been happening annually for the last 5 years or so, and it is a great place to talk with other modelers in the field, as well as people involved in open science. And we'd be very excited to interact with you there. And we will provide a travel stipend for one member of the winning team or teams to attend this Congress and present their method. In addition, all of the teams that stick through the challenge to the end and do submit a model to the final scoreboard for evaluation will be invited to join us on the final manuscript as well as part of the AD1 Challenge Consortium. So we'll officially call out as authors those people who are on the top teams, but everybody can be involved as part of this bigger consortium. There will be a variety of other smaller incentives to keep the game interesting along the way, which will be announced as we go. But this is the end game. This is what we're looking at as incentives for the final models that are submitted in September. So I'm, again, very pleased to have all of you involved. On behalf of DREAM and Gustavo Stolovitsky, who runs DREAM, and all of us on the organizing team, thank you. And so with that, I'll turn it over to Mette and to Simon to start talking a little bit more about the details of this particular challenge.
Thank you very much, Lara. Let's see here. I'm delighted to have Simon Lovestone here today. So Simon, I'm going to turn it over to you.
Thank you, Mette. And so hello, everybody. I can't tell you how delighted I am to see this happening and see some of the data that we've spent.
So I don't know if either Simon or Mette
collecting being used.
Oh, Simon is not on mute, but I can't hear him. Are you talking, Simon?
I was talking. Can you hear me?
Oh, now we can hear you. Yes.
All right. So apologies for that, everybody. I was just saying how delighted I am to see this process underway now and to see the data that so many people have spent a lot of time collecting being used in this way. It's really exciting. So let me start by just saying a couple of words about Alzheimer's disease. I know everybody on the call will know this, but it is important to remind ourselves this is a really big challenge. Alzheimer's disease and related dementias consume something like 1% of the world's GDP. It affects huge numbers of people, causes devastation to families and most important individuals. We currently can't do anything about the disease other than symptomatic treatments, but the world is moving fast and we're getting closer to the time when we can do real disease modification trials to look for therapies. But that work is being held up at the moment because of the lack of good biomarkers. So in Alzheimer's disease, we have both a challenge and an opportunity. The opportunity is that the disease process seems to take place at least 10 years and possibly even 20 years before there are clinical symptoms. So that's a real opportunity for treatment, because if, of course, we could treat people in that period, then we would have in effect a preventative strategy. But it also provides a challenge. And the challenge is we currently can't easily detect people in that preclinical state. And so that tells us we need biomarkers, and we're making progress towards biomarkers. But I think that what Synapse and Sage Bionetworks are doing in this Big Data Dream Challenge, the first of what I hope will be many for Alzheimer's disease, is to help us to deliver biomarkers. So let me just spend a couple of minutes to talk you through these three sub-challenges. So the first is to predict changing cognitive scores after the initial assessment. Now, as a clinician, I know that when I see people in my clinic, I know that there's going to be a range of responses over the next 2 years. Some people will progress very rapidly, others will progress very slowly. I can't tell from baseline measures right now which category an individual will fall into. If I could tell, then that would have immediate clinical importance, but even more importantly than that, it would help clinical trials, because if we could predict from baseline how people would progress, that would enable us to select and stratify patients in clinical trials, which is what we need to do for disease modification therapies. And the first sub-challenge is addressed to that challenge. The second sub-challenge is, can we predict which individuals are going to be cognitively normal even though they have biomarkers of amyloid change? So obviously amyloid is a key pathological process in Alzheimer's disease, and it is a puzzle to the community, to the scientific community, why not everybody with amyloid deposition in their brain has full disease. So some people have amyloid deposition without apparent ill effects. Other people have clear-cut dementia. Some people go on to decline quickly and others go on to decline slowly. So the second of the sub-challenges is to look in the ADNI data again to see whether you can identify from the data that's made available which of those people who are going to have biomarkers suggestive of amyloid deposition but remain cognitively intact. The third sub-challenge is to take baseline MRI, so magnetic resonance imaging data, and classify individuals into diagnostic groups, AD, MCI controls, and also to classify people into cognitive impairment levels by MMSE score. So this is of real importance clinically, but again, it's of huge importance for developing disease modification therapies and for the conduct of clinical trials. So these are three real questions. They're questions that the scientific community so far has struggled to find answers to. They're questions that might be addressed by the complex data sets that are being made available in this DREAM challenge. There are questions where it might be that there's signal amongst the noise of the complex data that's being made available. And I think if the people that are entering this challenge can find an answer to these three really important questions, then they will have done a really important thing for the field, as well as being a pretty cool way to do science. So I'm just delighted to see this happen. I really hope that Sage Synapse go on to produce more challenges in the Alzheimer's disease area, and I think that we can make even more exciting data available to those challenges in the future. And I just want to wish you all luck, and may the best team win.
Thank you very much, Simon. So before I turn it over to the two challenge leads, Keone Cowie and Arno Klein, I'd like to talk a little bit about logistics, about data and timelines and how you can get help. As Simon pointed out, one of the goals of the challenge, or the goals of the challenge, is to identify accurate and predictive Alzheimer's disease biomarkers based on this clinical genetics and bioimaging data that we are providing you. We hope that an outcome of this challenge will be tools that can be used by either the scientific, industrial, and regulatory communities. But do keep in mind that we have labeled this as AD challenge number 1. So as Simon stated, we hope that this will be the first of a series of challenges where we may refine the questions and also refine the data going forwards. Not going to go into much detail about these questions since Simon covered it already, and the challenge leads will do so even more so, but I want to point out what data sets are used for each of these questions. So for sub-challenge number 1, or question number 1, the training data comes from the Alzheimer's disease neuroimaging study, and the test data comes from the Rush Alzheimer's Center religious order study and the Memory and Aging Project. The same is true for question number 2, training data from the ADNI study, test data from ROSMAP, different subsets of those data, of course. And then sub-challenge number 3, the training data again is the ADNI study, but the test data here is from the European AD-Neuromed study. So just a little bit about the challenge timeline. We opened the challenge on June 2nd. By now, all the training data has been released. I'm going to show you through a screenshot where to access that training data if you don't have access to it already. In late August, we will be releasing the final test data that you will be making those final submissions based on. And we estimate that the challenge will be completed by September 19, and that's the final submission deadline. During this active challenge phase between now and the mid-September, the participants will have access to a leaderboard which they can submit their predictions and receive real-time feedback on the predictive power of your models. This leaderboard will be open by July 1st. We will record a instructional video of how to use this leaderboard, and we will post that on our Alzheimer's, on the AG Challenge site with detailed instructions exactly how to go about. And of course, we're available also for any questions that you may have. So again, the data for questions 1 and 2. Training data comes from ADNI, that is true for all 3 questions. It consists of genetics, imaging, biomarkers, demographics, and clinical data. The leaderboard test set for questions 1 and 2, as I said, religious order study and the memory and aging project. And what we will provide you with is an initial set that will be used for the leaderboard and then a final test set based on this data. The answer, we also have a part of the AD-NeuroMed study. This is part of the data that does not have the imaging component, but we're providing that through Synapse as an ancillary data set that might be useful for addressing question number 1. Now, I do want to make the point that you're welcome to use any ancillary data that you wish in order to answer these questions. You're not restricted to what we have provided you access to. The data for question number 3, ADNI data again for the training set, and this set consists of genetics, imaging, demographics, and clinical data. And the leaderboard test set and the final test set comes from the AD-NeuroMed study. Imaging, genetics, and clinical data here as well. So how do you get to this data? How do you access the data? For the ADNI data, these data sets are accessed through the ADNI database, the LONI Imaging Archive. And in order to get access to this data, and there's, if you see the link at the bottom of this slide, we have very detailed instructions, but the initial step is to agree to the ADNI data use agreement. You're then sent an email that will give you a login to the LONI image database. For the ROS-map data and the AD-NeuroMed data, both of these data sets are distributed through Synapse. For the ROS-map data, we require that you submit a document, a data use agreement, where you must agree to certain terms of use in order to get access to the data. And again, the same with AD-NeuroMed, you don't have to send in a document for that, but there is also a requirement that you must agree to certain terms of use in order to being able to download that data from Synapse. So here is where, once you have access to the data through LONI, this is where you find it. For the clinical files, for all 3 questions and the imaging files, you go to download study data, and the section, the tab where it says test data. This is, keep in mind, this is a training data set. It's not the test data, but that's how it's been labeled. And the 2 files that you want to download is the AD Challenge training data for clinical and imaging, depending on which question you want to answer. For the genetic data, you go to download genetic data, and again, there's then a tab that says test data, and these are the imputed genotypes that we have generated specifically for the challenge. Keone will talk a little bit about this, but we have imputed the genetics data across all 3 data sets using the same parameters. So we're providing this imputed data for you. Now, once you have access to the ADNI data in LONI, you have access to all the ADNI data in LONI. You're not restricted to what we have provided specifically for the challenge. So I do want to point out that there is whole genome sequencing on part on ADNI1 and part of the ADNI data, and you also have access to the raw genotypes, not just the imputed genotypes that we provided. Okay, so here is a screenshot. Chris will show you live how to find this data in Synapse, but I just provided a screenshot right here. So in order to get access to ROSMap, you need to download a data use certificate first. That can be found under the Files tab under the Resources folder. You need to have this certificate signed by yourself and an institution signing official, and then submit that to us at the email address indicated on the slide. We'll process your submission, and if everything looks okay, it's not a complicated process, you are authorized to download the clinical and genetic data files from Synapse, which can be then found in the ROSMap folder labeled Leaderboard Q1 and Leaderboard Q2, depending on whether you want to participate in one or both of the questions. The Add Neuromed data, again, this is accessed through Synapse. Here, you don't have to submit a data use certificate, but you do have to, and please read these terms of use, you have to accept these terms of use before you can download the data files. Now, what
Uh-oh, Meta seems to have cut out. Meta, are you there? Okay, well, perhaps I'll take over for Meta. Stepping right in. So the point here, not to belabor this point, the point here really is we just want this to be a fair challenge. So if you've used this data or viewed this data before, we ask you to recuse yourself from the challenge. Meta, can you advance the slides? We might actually need to just change the, give me a second. I have, finding the slides. There they are.
Change presenter.
Yeah. Okay, so we'll change the presenter to me. And sorry, we're having just a minute of technical difficulties. Okay, here we are. So let me just show my screen and find where we are in this. Here we are. Okay. I think we are back on track now.
Yes.
Okay, great. So this challenge has leaderboards affiliated with it, and so this is an exciting opportunity for all of you to get real-time feedback on your models as we go through. So the leaderboards are going to launch soon. The leaderboards for questions 1 and 2 should go live next week, and then for question 3, hopefully not too long after that. So the way the leaderboards work are that you will be building your models and the training data, and then a subset of either the ROSMap data for questions 1 and 2 or the Add Neuromed data for question number 3 will be used as a test data set in the leaderboard. And so you can submit predictions for these 2 test data sets, and you will immediately get your scores. Or they will be immediately scored, and both your score and your rank relative to other submissions will be posted on a public site in Synapse on the challenge website. And so you can view how well you're doing. There'll be more information on the logistics of exactly how to do this as these things go live over the next week or 2. My slides are not advancing. Oh, there we go. Okay, so how do you get help? Well, there's plenty of ways to get help. We are here to help you. A really great resource is the Challenge Wiki. A lot of work has gone into making sure that all of the information we have anticipated you would need is up in the wiki. So please take a look up there. There should be both information on scientific content as well as logistics of how to make this all happen. So that's a great place to start. However, if your question is still not answered there, we also have a forum. And you can find information about the forum at this link here. You can also find it from the Challenge Wiki website. And on this forum, you can answer a question or you can ask a question, and we are constantly monitoring these, and we will try to get back to you as quickly as possible. With the caveat that next week, many of the organizers of this challenge are going to be on vacation. So next week, you might get a slower response than you will otherwise. We will try to answer questions within 24 hours generally, but next week there will be a little bit of a lag time. We do not encourage you to email us questions independently just because so many people have the same questions. If you have a question, chances are other people do too. And so we would ask you to put it up in the forum. However, if somebody does email us a question, we will not email the response back to them. We will paste their question into the forum and answer it there. And that way everybody has equivalent information across all participants. Okay, so let's go into a little bit more detail about each of these sub-challenges and what they entail. And with that, I'm going to turn this over to Keone, who's one of the challenge leads and has been working hard to prepare questions 1 and 2. Keone, take it away.
Great. So, I mean, Simon did a wonderful job of introducing the context
I
of
don't
these
know
questions.
if you're talking, Keone, but we cannot hear you.
I am talking and I'm not muted. Can you hear me?
Oh, we got you.
Okay,
Thanks.
maybe there was some lag time there. So Simon did a wonderful job explaining the context of these challenge questions, so I won't spend a lot of time on that. But he did talk about the opportunity for early treatment and prevention and the challenge that we need to come up with ways to detect that early pathology or that preclinical pathology. And that's really part of what this challenge is about. So we have a large, we have the sample set up. We're looking at changes in a cognitive score called the mini mental state exam over a 24-month period. Some of the individuals are initially non-demented and become demented. Others remain non-demented throughout the two-year period. And the challenge is focused on being able to develop biomarkers using both clinical and genetic information that can predict this trajectory. The models that are developed during this challenge will help us leverage the opportunity that Simon talked about. We can have more efficient selection of samples for clinical trials. That means they can be run with smaller numbers of individuals for shorter amounts of time, but still be just as effective. They also open up possibilities for earlier disease treatment if those biomarkers give us some information about the fundamental aspects of the pathology of disease that could provide important insights. So that's how this question leverages the opportunity and challenge that Simon talked about. Can we go to the next slide here? Okay. So the data in a little bit more detail. The training data come from ADNI, as Meta mentioned, 767 participants. The primary phenotype of interest is the delta MMSE, which is being defined as the 24-month MMSE score minus the baseline score. And this is a score that ranges from 0 to 30. Other measures are provided, including clinical diagnosis at baseline, age at baseline, years of education at baseline, gender, ApoE4 genotype, that's a major genetic risk factor for disease, and whole genome imputed genotypes. And I'll talk in a little bit more detail about those, but you can see we have worked really hard on the wiki, as Laura mentioned, and you should have a very detailed explanation of the ADNI data and the variables and the format of the data files by following that link. The ROS map study will be used for the leaderboard data and the final test data. It's been randomly split into those two pieces of 588 and 587 individuals. As you can see, the final test data will be released in late August. And again, the measures provided are baseline MMSE, diagnosis at baseline, age, at baseline years of education, gender, E4 genotype, and whole genome imputed genotypes. And the only thing that is withheld, of course, is that 24-month MMSE, and that's what needs to be predicted. So the basic timeline here, June 2nd is when the training data and leaderboard data were released. The leaderboard will be open on July 1st. The clinical features of the final test data set will be given out in late August. The time period from August 22nd to September 1st is the time period for the final submission for the clinical predictions using just the clinical data. And September 5th is when we will release the genotype data. And then models can be built using both clinical and genetic data, and those final submissions will be accepted from September 5th through September 19th. Now, I just wanted to touch on the genetic data and make sure that everyone understands how it was collected and what we have provided so that you can think about that in terms of how you want to leverage it for your models. The ADNI data was genotyped in 2 separate phases. Some of it was genotyped using the Illumina 610 quad chip. Other part of it was done using the Illumina Omni Express chip. What we did was we imputed each of those chips separately to the latest, to the HG19, and then merged those data sets and cleaned them. So on the wiki we have a lot of detail on the imputation process, and we won't go over that during the webinar, but we have every aspect, every command that we used for the pipeline, including prephasing and shape it, imputation using impute2, and then some post-impute QC using QC tools. So you can see the same format here for ROSMAP and AdNeuroMed data sets. We have different chips, and we've imputed them and then consolidated them, cleaned them up, and prepared them for you. So that's the data and the context there for sub-challenge 1. So for sub-challenge 2, we have a really unique question. This is a question that I personally am very interested in, and I hope that this challenge will yield some really great insights into this. So there's a very interesting phenomenon, and that is that a large number of elderly individuals who die non-demented, when you perform an autopsy on them, their brains actually have full-blown Alzheimer's pathology, despite the fact that they have no evidence of any cognitive decline or cognitive dysfunction. And so this challenge is really about taking these unique individuals who show evidence of amyloid pathology in their brains, but show normal cognitive function, and trying to understand what the biological basis of this resilience actually is. What is allowing them to be robust and resilient to this pathology? And you can see pretty quickly where this could go, right? If we can understand how people are resilient to the presence of Alzheimer's pathology, we have some real insights into the types of therapeutic targets that will help us prevent Alzheimer's disease. So to me, this is a really important challenge. And it's a very unique subset of individuals, and understanding them would have a great impact on the field. So scoring here, you submit one prediction using both the clinical and the genetic information. Submissions can be put in at any time, but each group will be, or each team will be limited to 100 total submissions. For this final submission, there'll be one phase using both the clinical and genetic information, and you have 2 predictions. So once that final data is released, you have two shots at your best model. For the scoring metric, predictions are going to be assessed using the area under the ROC curve and classification accuracy, and so a combination of the performance based on those 2 measures. Now, so the data that's available includes 174 cognitively normal individuals and their assessment at baseline, similar assessments to what we saw with question 1 in terms of clinical data. But the primary phenotype is discordance, and this is discordance between the indicators of amyloid pathology and cognition. In this case, what we have is a subset of these cognitively normal individuals who have measurements of amyloid in their cerebral spinal fluid that indicate the presence of amyloid deposition. So you have this discordance variable along with other measures including MMSE, diagnosis, age, years of education, gender, E4 genotype, and all the SNP data. And you'll be using that to build models that predict whether someone is showing evidence of being discordant or having normal cognition in the presence of pathology. Again, you have a link there to the wiki that has a detailed explanation of including headers of all the files so that you can understand what each variable means and how you might leverage it for your models. The test data is ROS-map. We have 129 cognitively normal individuals, and their assessments of baseline. The discordant status is known based on pathology, so they've gone to autopsy, and we have data from the ROS-map study on their plaque and tangle counts and their Alzheimer's diagnosis, along with the appropriate variables that match there and details at the wiki. And again, we've held back half of the ROS-map data set to be used as the for the final scoring. So basic timeline, the data have been released. The leaderboard opens July 1st. We'll release the final test data September 5th, and you'll be able to put your two best guesses in between September 5th and September 19th, or your two best models. Submission format for this particular challenge has been finalized, so we have that here for you. And you can see we'd want 4 columns named project, PROJID, rank, confidence, and discordance. So the ID is the pre-ordered list of subjects. I'm sorry, the ID is the identifier. The rank would be your pre-ordered list of subjects from most discordant to least discordant. And then for each subject, you would also have your confidence in that ranking. And then finally, a binary prediction of either discordant or concordant. So we're excited about these two questions and hope that you will be able to come up with some fantastic models that predict cognitive decline or cognitive change over time and resilience to Alzheimer's pathology. So thanks.
Now, thank you very much, Gioni. And now for sub-challenge 3. What distinguishes sub-challenge 3 is that we're going to be making use of brain imaging data. The challenge itself is to classify individuals into separate diagnostic groups. That would be Alzheimer's disease, mild cognitive impairment, and healthy individuals, as well as to predict a cognitive score, mini mental state exam cognitive score, using magnetic resonance imaging data. And as Simon Lovestone said a little bit earlier, the theme here is to try and find biomarkers. And in the case of structural MR data, the natural biomarker would be morphometry or shape-based data. The rationale for doing this is that if one could, with a single MR image, differentiate Alzheimer's patients from those with mild cognitive impairment or healthy individuals, we can focus research on specific anatomical structures or regions of the brain that are different between the groups. The data, as Meta mentioned earlier, consists of a training set from ADNI and 2 test sets, a leaderboard test set and a final test set from the AdNeuromed study. They include MR data, information about age, gender, years of education, APOE4 genotype, and whole genome imputed genotypes. Now, you are welcome to download all the MR imaging data that you care to. For your benefit, we've conducted a lot of processing on these data. 3 pipelines. One is FreeSurfer, the Recon All pipeline, which is used for extracting cortical surface meshes, tissue class segmentation, and labeling. The Advanced Normalization Tools, or ANTS, the ANTS cortical thickness pipeline, which produces nonlinear deformations or transforms that take individual subjects' data into a template space. And finally, MindBoggle pipeline, whose website is mentioned there. And what MindBoggle does is it takes the outputs from FreeSurfer and ANTS, creates a hybrid segmentation, propagates the labels through each of the tissue classes, in those volumes, as well as extracts features such as sulcal folds and measures shapes in those folds and labeled regions. Now for each vertex or point on the cortical surface, at bottom you see 3 different images of the same brain. It's about 140,000 vertices per hemisphere's mesh. In the bottom in the middle, you see a depth map where each point in the brain has a color code, which is some shape measure, in this case, travel depth. To the left of that is a partially inflated surface with a curvature, a mean curvature map. And on the right at the bottom, we see a different depth map for just folds in the brain. So the shape measures for each of the cortical surface vertices include such things as surface area, travel depth, geodesic depth, mean curvature, free surface measures of convexity and thickness, cortical thickness. And for every labeled region, we can compute volume, as well as Laplace-Beltrami spectra. Now, given that there are 140,000 or so vertices per hemisphere's mesh, that's a lot of numbers to deal with. And so oftentimes what we do in MR data, whether it be structural or functional or otherwise, is we delineate the surface into different brain regions. So here we've done this for you. Each cortical hemisphere consists of 31 labeled regions. And so the labels are either the cortical surface brain regions, non-cortical brain regions in the volume throughout the brain, or folds extracted from the brain. The shape tables from these image process data take 3 forms. The first is in a table called vertices, you'll find in the subfolders. They consist of shape measures for every cortical surface vertex. So if, for example, for the left hemisphere, there are 140,000 vertices for a given subject, you'll have 140,000 rows. And each column will give some of those shape measures: depth, curvature, convexity, and so on. Also, for the labeled regions in the whole volume of a brain, we have a second table called free surface filled labels _ volumes, another comma delimited table, where, simply put, for each labeled region, you have a volume. And finally, we have cortical surface shape statistics, and that is for every labeled region in the cortex, or for every sulcus fold extracted from a cortical surface, we have a table like label _ shapes and sulcus _ shapes, where every row is a labeled region or a sulcus fold, and every column is a summary statistic of those, say, thousands of vertices for a given labeled region or sulcus fold. And those summary statistics are a range of different features, from the usual median absolute deviation, mean, standard deviation, kurtosis, skew, lower upper quartiles. And so there is some repetition in there, but we didn't want to presume to know what participants in the challenge would like to make use of, so we make them all available. For scoring, we want you to predict 2 things. One is which diagnosis group, that is Alzheimer's, mild cognitive impairment, or healthy controls, as well as can you predict the mini mental state exam score. The reason that we actually pulled the score out of the diagnosis group is because it's one factor in the diagnosis. And it would be interesting to see how well you can predict that. For the leaderboards, both predictions will be in the same submission file as in the example on the right, where we have columns with ID, the MMSE score, and diagnosis. You can submit anytime, up to 50 times. Be careful of overfitting. And for the final submission, we would want you to do the same thing, predict MMSE score and diagnosis, but you have only 2 chances in the final test set. And the metrics for scoring, we're going to have a Pearson correlation of the predicted MMSE against the measured MMSE scores and % correctly classified subjects. And we're going to take the average of those two scores, and that's what's going to make it up to the leaderboard and the final rank. Finally, the timeline. The training data have been released as of June 12th. The test data will be released. We're not sure it's going to be on the 27th, but we're going to try our best. July 1st, the leaderboard opens, as was mentioned earlier. September 5th, the final test data will be released, and up to the 19th, the final submissions will be accepted. Thank you very much. I really look forward to your participation in Sub-Challenge 3, and I look forward to answering any and all further questions, if not in this forum, in the forum online.
Should I turn it over to you?
Yep.
Okay. Thank you very much, Keone and Arno. So the last piece of this is that we're going to provide you with some information logistically about how to use Synapse to participate in this challenge. And this is going to be presented by Chris Baer, who is an engineer on the Synapse team. Let me just change the screen over to him.
So while she's doing that, like Laura said, my name's Chris. I'm a software engineer on the Synapse team. And if I click show my screen, I think shortly we should be able to see Synapse. So this, for those of you who haven't used it yet, this is Synapse. And I'm just going to walk through some of the ways that you as participants will be interacting with Synapse. If you don't have an account already, the first thing to do is register, which happens right here. This is very simple, same as any other website. The one thing to know about this is that within the challenge rules, you do have the option to participate anonymously. And the way you do that is just don't fill in your name here. These say optional because they are optional. Your username will be publicly displayed, so choose that in a way that if you choose to be anonymous, we'll preserve that. Your email will not be exposed publicly, so you can use any email you already have. We do, however, kind of encourage people to participate publicly and be proud of their participation in this challenge. So we definitely encourage you to fill in your information, but anonymity is an option here. I already have a little account set up, so I'll quickly log in here. Oh, I don't want to save that password. As a new user, this is what you'll see, kind of a blank slate. Down here in this carousel, you can find your way to the challenge. And one thing that is convenient to do is to click this little favorite icon here. Now that shows up in your favorites. It makes navigation slightly easier. The next thing you're going to want to do, quite possibly, is to create a team. And that's in this little box here. I'm going to name my team Turbo Modelers. And if you create your team, you can invite people to join your team. Maybe Arno wants to join me here. So that's how you would do that. You can set up a whole team, invite your team members. I won't go through the rest of that process. So now that you've registered for Synapse, you have found your way to the challenge, created a team potentially. The next step is going to be accepting terms of use of the data, which Meta already went over. Actually, the next step is joining before you do that. So just click on this blue button here. This process will walk you through the kind of restrictions and legalese involved. The Dream Challenge rules specify your rights to access the data, what the limitations of that are, things about publishing, so you'll want to be familiar with those. And you'll accept those. And you are now a member of the challenge. Just to point out that in your profile, it will show that. So I'm a member of the team Turbo Modelers that I just created, and I'm also a participant in the challenge. Another thing you're going to do is to create a project. The project is going to be kind of a home for your models, your code, your data, and also wiki write-ups. So you can create a new project right here. My 80 challenge. So the project has this component files and another component for a wiki. I did want to kind of demonstrate a couple of the interesting features of the wiki by showing some projects that were from a previous challenge. This is from the Toxicogenetics challenge, and these were some of the really nice write-ups that people did. And if you guys are on the call, hello. These guys did a great job. They illustrated their method with figures, heat maps. They described each of their steps in their workflow, gave you a nice LaTeX equation here, and a tabular view of their different submissions and how they were ranked. You might also do like how they were parameterized, which steps that they used. So we really encourage kind of a nice write-up. These make a nice summary of your methods that you can show to other participants in the challenge.
And I'll just add in that when you submit your final predictions, the winners will be required to provide both a write-up and open code. So it's not just encouraged, it's required in order to be selected as a winner.
In order to kind of help you along with that process, we've created a write-up template. This sort of shows the sections that you might want to have in your write-up. And then lower down, it kind of shows a bunch of the different features of the wiki markup language that you can use. It's a very simple way to include figures, data provenance, equations, and all kinds of good stuff into your write-ups. So this will be available to you to use as a template. Yeah, next thing I want to talk about is how to submit results. Let's see. I'm just going to do this programmatically first. Or sorry, I was going to do this on the website first. So this is kind of the template. You might have a folder structure that looks something like this. Within your project, you'll have folders for questions 1, 2, and 3. I've uploaded my glorious model here, which was created by a random number generator. You guys will do much better than I have. And if you go over to this tools menu and click submit to challenge, you can say which of the questions you're submitting to. I'm going to say question 1A. I can name my submission totally random. And my team name was Turbo Modelers. I think I had a capital O, so I'm gonna continue my typos here. And I would just hit OK to submit. I'm not actually going to do it, but that's the submission process on the web client. Synapse also has an API that is available from Python and R, so you can do this thing. You can do the same thing programmatically. If you can read this code here. I'm going to just quickly go through. I am a software engineer, so I have to show some code. This is the Synapse Python client, importing the libraries, logging in. You'll actually put a username and password the first time you do that, and it caches an API key. So you don't actually have to expose your password in your code, which is a nice feature. This is creating a project. You probably will have already done that, so you'll just get the project from Synapse. You can create folders programmatically. You can, if this is a file on your local file system, you can upload it into Synapse. It becomes a file in that folder structure. this get evaluation thing is the way that we map submissions to the question. There's a table in the project wiki that tells you which IDs to use. So basically, you're putting this in a queue, and some code will validate and score your submission. This ID is kind of the key to finding the right queue. And finally, you submit. So you say, I'm putting into this evaluation queue my predictions, and just like I did on the web, you give it a name and a team name, and you've made a submission. The submissions also have an ID, so if you want to keep track of that, it's a good idea. It comes in handy when asking questions later, particularly sometimes a file format issue or something will come up, and it's useful to refer to those submissions by IDs. I think that's very nearly all I have. The one other thing I wanted to put in was, I believe Meta already mentioned the links to the support forums. If you do ask a question, it's not required, but I'm just going to type a question. What is up? The forums we use have this concept of a product. So the product, quote unquote, here is the Alzheimer's disease challenge. If you remember to check this, it will get where it's going faster. If you don't remember, no problem, we'll find it. But this will make it a little more streamlined. So I just wanted to point that out. And I think that's everything I have.
I just was wondering, Chris, if you could explain to them what to expect once they've submitted
Sure.
to the leaderboard or to any of the evaluations.
Yeah, so the submission process goes like this. The submission goes into a queue. There's an automated script that validates that it's in the correct format. You will get an email that either says, yes, we've received your submission and it looks good to us, or we couldn't read it for whatever reason, perhaps the wrong column names or IDs or something like that. So you'll get a validation email. It will go into a queue to be scored, assuming it's valid, and you'll get back the statistics for that particular question. So you'll get back an email that will give you the score as well. It will appear on the leaderboards, which are forthcoming in a week or so.
Great, thank you. Okay, so that's the crux of it. That's what we've provided for you. Just one final note to say there's a lot of people involved in organizing this challenge. It's complicated. We have multiple questions. We have many data sets involved. And so a thank you to all of these people. And many of these people are online to answer questions online to answer questions as we go along. There are also many other people who have been involved conceptually in this project, and so they are listed here. And so a shout out in particular to our data contributors, Rush University, ADNI, and AdNeuromed, which is represented on the phone by Simon Lovestone, as well as our funders, sponsors, and our general partner. So at this point, we're going to open it up to questions. So please, if you have a question, please type it into your question section, and we will go through them one by one and answer them. So the first question is regarding data use. So we had said that so the question is, as far as I understand, we can use any data from ADNI, in which case does this mean a team is disqualified? So let me clarify. We are disqualifying teams that have previously accessed the test data sets because they have an unfair advantage. And the test data sets are ROSMAP and AdNeuromed. So the ADNI data is purely for training, and so you can use whatever data you want. Just keep in mind that not all the data that's available in the wider ADNI database is actually available for the test data. So be careful with what parameters you select, because you may not be able to use them in the test data. Okay, so the next question I believe is for Keone. So I'll read the question and hopefully Keone can answer it. For challenge 1, the models will be evaluated by an average of Pearson and Spearman correlations. These are unusual scoring methods. Why are these more exotic scoring metrics used instead of a traditional metric such as the root mean square error? Keone, are you there?
Okay, can you hear me? Yes, I'm here. Can you hear me? Hello?
Ah, there you are.
Okay. So yeah, we certainly talked about a number of different metrics for scoring, and we tested several metrics as we did some dry run models, and we really felt like this particular combination of metrics captures um, several different aspects of the models that we wanted to capture, and so they are a little bit more complex, but they do allow us to more thoroughly evaluate the models and get a little bit better feel for the way that they predict the actual scores. So, I mean, there's not a simple answer other than we felt like this combination of models better captured the accuracy and strength of the predictive models than other options.
Great. The next question is an easy one to answer. The question is, can we obtain copies of these slides? The slides will be made available through the challenge website, and we will send out an email that provides you with a link to the slides, as well as a link to the audio or the visual recording of this presentation. The next question is by an individual who's having problems downloading files from the ADNI website, and I'm having problems downloading them. Yeah. So I guess we'll have to follow up with Rintu offline. I'm not sure exactly what the issue is, and I hope it's just an individual issue and not something more systematic.
If it's the imaging data, those data are very large. And actually, we had a little bit of difficulty ourselves with Lonnie's image data archive, downloading it all in one go. So it does require some persistence. And it also did not work on all browsers. So if it's the imaging data, remember these data are very large, and to maintain a connection and get it all on your computer may take a bit of work. And that's the reason why we took pains to process those data, because we didn't expect that everybody would have the resources, not just to download, but to crunch the data and analyze.
So the process data is not that large, but the original imaging data is.
Yes.
Yeah. Okay. So if that's not what the problem was, Rintu, then please, I guess, if you could follow up with us through the forum, we'll help you out with whatever way we can. The next question is a clarifying question about the data. What's the difference between the leaderboard data and the training data, and which are used for model building and evaluating the test data? Okay, so the training data in all cases comes from ADNI, and you will have both the features that you will use to build your models, as well as the variable that you're trying to predict. So you can use this to develop your model. The leaderboard data comes from a totally different study. So for questions 1 and 2, it comes from ROSMAP. For question 3, it comes from AdNeuromed. And in this case, we will provide you with just the features that you would use to predict, so you can build your model in the training data and then apply it to the test data, upload your predictions into the leaderboard, and get real-time feedback as to how well you're doing. So you won't ever actually have the phenotypes that you're trying to predict for the leaderboard. You'll only be able to use that to get a sense of how well you're doing. The final test data is a completely separate holdout of those data sets, the ROSMAP data and the AdNeuromed data, and so it's essentially a third data set or a second validation that can be used to assess final models. And this is so that anyone who might be overfitting to the leaderboard won't have an unfair advantage in selecting final models.
Also, I'd like to add, with respect to the sub-challenge 3 and the imaging component, that the two test sets, the leaderboard test set and the final validation test set, the reason that was also split into two is because the ADNI data and the AdNeuromed data, though the AdNeuromed is the European counterpart of ADNI, they drew from slightly different populations and they use different scanning facilities. And so we expect that given the difference in demographics, I think younger, different education level and all, and different possible artifacts or scanning parameters, that it is a different sample. And so as closely as they try to match each other, we can expect a full carryover of a model. So the model should be somewhat robust. So after full training with, say, series cross-validation, the leaderboard test set is intended to be able to get as close as you can to a final answer. And then the second test set, the final test set, is intended for your final submission.
Thanks. The next question is about the ADNI data, and it's a clarifying question. Is there raw individual genotype data available elsewhere in ADNI? Because what we're providing you with is the imputed dosages. I believe the answer to that is yes, it is available, but I wonder if either Keone or Meta could confirm that.
Yeah, I'll come on. Can you hear me?
Yes.
Hello? It is absolutely available. Individual level data from the two separate chips, as well as VCF files from whole genome sequence for a subset of the individuals.
Thank you.
So if you go to the genetics tab, when you get into the ADNI website, because they do have access to all of it once you get in, you should be able to find that raw genetic data, including the VCF files from the whole genome sequence, independently of the SAGE challenge data sets.
Great. Okay, the next question is, when will access to the IBM server be available? That's a great question that I don't know the answer to. I imagine it might be available now. Does anyone know?
Would be one for Gustavo.
So we'll answer that question on the forum. We'll need to confirm. Unfortunately, we lost sound with Meta because I'm sure she knows the answer.
Actually, can you hear me?
Yeah.
I got sound back. I apologize for the disruption of losing my sound here. I'm on a rather unstable network. Yes, so the IBM servers will be available. We're just working out some final kinks in the process concerning getting access to the servers, but they're very happy to provide it, and we will put an announcement on the forum as well as in a weekly newsletter, email newsletter that will be continue to send out.
Excellent. Okay, the next question is, for the training data, can we use extra biomarker data in ADNI to build our models as ADNI is open access? Absolutely, you can use whatever you want in ADNI. Just be aware that that information is not necessarily going to be available in the test data when you go to predict there.
If I could add to that, in the imaging component, the question may come up for those who are familiar with ADNI. It has a wealth of different imaging modalities. The AdNeuromed does not. So we had to restrict the data used from ADNI to match those in AdNeuromed. So you're welcome to train models that would use everything from PET, fMRI to fusion, whatever you care to. Again, as Lara said, realizing that you will not be able to make use of those data from the AdNeuromed set.
Thanks.
Let me add to that also. I think there are ways to leverage all of the data. So you just need to basically come up with ways that the variables that are present in both the training and the test data sets can be used to represent the broader data from ADNI. So I think there are ways to leverage it depending on how creative you are about relationships between the variables.
That sounds like an open challenge all in itself. The next question is, can I work on a personal desktop? Absolutely, you can work wherever you'd like.
Yes, it's a Commodore VIC-20.
The next question is, will the teams from the 3 sub-challenges be measured separately or ranked all together? They will be measured separately. The next question is, will you release the leaderboard data with the answers for training of the final model? And the answer is no, we will not. And that is the last question. Okay, so with that, I think we're going to wrap up. Again, if you have any other questions today or at any other time in this process, please ask them on the forum and we will continue to answer. Happy modeling to all of you and best of luck. Thanks for joining us today.